Hello everyone, welcome back to BioBoost. Today we are going to cover transcription topic. In my last video, I discussed about replication. This process takes place once the DNA is replicated, and now we need to make from the DNA strand one RNA strand. We will cover prokaryotic transcription, where we will learn about RNA polymerase, the origins and promoters, and the three phases. Also eukaryotic transcription, the RNA polymerases, and the modifications which take place once the stranded made. So what is transcription? It is the process where a segment of DNA is copied into RNA by the enzyme RNA polymerase. Here you can see the strand of mRNA being made from DNA. Here, after being processed, RNA exits from the nucleus and enters the cytoplasm. It is the processed mRNA. The difference between the transcribed mRNA of prokaryotic and eukaryotic you can see here. It is a direct process, but in eukaryotes, it undergoes a process named as splicing, which is very important, which we will cover next video. Here you can see the processed mRNA. Now, we will discuss about the main enzyme involved in transcription, which is the RNA polymerase, and it is the enzyme which catalyzes the synthesis of RNA on a DNA template strand. Here you can see on 5 end, the tail is outside. Only from the 3 end RNA is getting transcribed. Here the coding strand is there in 5 to 3 direction, which is same as of RNA except the T and U bases. But the RNA and the template strand, these both are complementary to each other. Now we will see the properties of this enzyme. So it is very important because it requires no primer for polymerization and it uses DNA as a template. It's active when it's double stranded. It's synthesized in 5 to 3 direction and requires magnesium ion for RNA synthesis activity. Its exonuclease activity is from 3 to 5, which is absent. Error rate is 10 to the power 4 to 10 to the power 5. Now here we can see the holoenzyme structure. It has 5 structures: alpha, beta prime, 2 alpha units and 1 omega unit. Their weights are given size wise. Their molecules are double of alpha unit and their function are very important in this whole process. Sigma is for promoter recognition. It is very important because the enzyme responsible for RNA synthesis is DNA dependent RNA polymerase, and it is multi subunit. A total of 480 kilodalton is its weight. Looking at the function of each subunit, you can see the alpha subunit is responsible for chain initiation and interaction with regulatory proteins. Beta is for chain initiation and elongation. Beta prime is for DNA binding and sigma is for promoter recognition. In next initiation phase, you can see that the sigma part get detached once the RNA polymerase recognize the promoter sequence. Here you can see the core RNA polymerase structure and how after addition of sigma factor it becomes a holoenzyme. Next are prokaryotic promoters. These promoter regions are from -35 to -10. Minus represent it is an upstream from the transcription start site. -10 one is the TATA box or Pribno box. It is very important. -35 is the transcription start site from which 17 base pair is the difference between these both regions, and it is the place where RNA polymerase binds basically. So this is very important from initiation perspective. From transcription start site, this one is the upstream and their downstream plus one is the starting site. Here you can see in various species of E. coli what are the promoter sequences are, and this is the 17 base pair distance between these two boxes. Next, we will see the transcription process steps. It involves promoter binding, DNA unwinding, initiation, elongation, and termination. The 3 steps are different for both prokaryotes and eukaryotes in the initiation and termination phases only. Elongation is same in both of them. Starting from initiation, here you can see RNA polymerase recognizes the promoter sequence and starts the transcription. It recognizes the -35 to TATA box region, -10. Then opens the DNA duplex between 17 base pair which are present between in these two sequences. Here you can see rewinding of DNA again once it is getting transcribed, and here unbinding of DNA takes place. It requires GTP, RNA transcript. It is always a purine phosphate. Formation of transcription initiation complex is made at initiation and no primer are needed for RNA synthesis. This is very crucial point. Sigma subunit falls off first after first bond. So sigma unit will be detached. Next is elongation. So here RNA strand will grow continuously, and because the sigma is getting detached, it causes change and it becomes a core enzyme. So it slides on DNA template to 3' RNA polymerase. Here free NTPs in this diagram you can see are added to the 3 OH region of nascent RNA and a transcription bubble is made. 3 segment of nascent RNA hybridized to DNA, while the 5' here you can see the tail region is extended outside the whole complex region. Next and last is termination. Here RNA polymerase stops on DNA and RNA transcript falls off. Termination either occur with this subunit, which is Rho-dependent or Rho-independent termination. Here you can see if it is dependent, then the Rho factor is a hexamer helicase which is involved in termination and it requires ATP for its activity. While the Rho-independent one, it involves a signal which is 30 to 40 nucleotide RNA, which is followed with a GC sequence and a stem loop structure. Here you can see Rho-independent termination and this one is Rho-dependent termination. Here, when the pre-mRNA is transcribed, a region of AU sequences are there. While in Rho-dependent one, hairpin or stem loop structure is formed, which is a GC sequence. Here you can see the bubble is made and the Rho-dependent Rho factor is bound on the DNA RNA hybrid complex and it forms a GC complex, which these are inverted sites which are basically complementary to each other and they bind to each other. So they form a hairpin stem loop like structure. Next we will see that after processing RNA and tRNA how these are made. So their biosynthesis. So once a pre-RNA transcript is formed, there are different sizes. We know after density gradient how this is made. So different mature RNAs are made like this. So this was